So he has to speak into the microphone and I would have to speak into the microphone as well.
Yeah, but it should hear you nicely.
So on this one,
Yeah.
it's automatically switched on.
Changed it into a holding structure.
Okay.
That's for every country separate company, separate institute. Moving to also impact project, Barbados, for example, so it's growing, we're going to start in the same area.
Yeah, okay. It won't be heard online.
Okay. Okay.
Yes, so University Paris-Saclay, and but we're going to be inside the incubator building of Sophie.
Can you please remember the picture maybe taken and you say
First project called Closes.
It's
about
Can you feel the room or?
hearing those.
Okay.
So most of the time, the two epitopes put it on an antibody, there's no way.
Yeah.
Very good. You never know, right?
What are you doing at Center?
So I'm the alliance manager, so I take care of, I work with you and
Ah,
for
there we go.
all the MOGSK projects, but also with the university. Now let's see what happens with the clients, about opportunities.
That should be.
So, yeah.
We should.
Yeah, we should, right? I thought afterwards, yes. Yeah.
I'm so surprised.
Right? So,
One stream from different directions.
Yeah.
From the car,
I know.
university, others from industry.
State, federal.
yes, and for us, it's a whole contractual big mess, so I talked to Andrea when they were trying to, they were drafting the contract. And so, yeah, money is great. So sorry.
No problem.
This is really cool.
Of course,
Yeah, so she's our boss,
I'm
and
sorry,
whereas Nazi is more in the alliance manager, I'm more looking on the scientific part. So she just started that role like half a year ago.
Are you going to be in Vienna at BioMed?
I have, say, 8 children at home and 4 children, so.
Of course, we have 2, but both left school. So, at home.
We made it kind of efficient. So we had one, and then we got triplets. And they just started school, so it's kind of a total mess. But anyway, the cool thing is there's so much need on the years, so there's still enough to see. Yeah, I'm curious to see that.
Ist der Coffee in der Kamp?
Yes.
Hallo.
Gut, gut.
Ja, ist das was?
Ja, ich jetzt. Doch, Hardtop. Also durchaus ein Grund. So, keine Gedanken. Ich habe man kann ja nicht sagen,
Also das war vielleicht.
Wow, alles gut, alles gut. Das ist ja fast peinlich, so bewusst.
Das ist toll. Ich bin begeistert. Wo? Also ich bin jetzt ganz warm. Gut.
Ich lese das nicht einfach vor, was in den BIOS
Nein,
steht.
nee.
Ich versuche einen Schwarzteil zu
Ruhe
machen.
bewahren. Das ist sehr simpel.
Ja, ja, maybe. Yeah. So, good morning, everybody, and a warm welcome on behalf of EMBL and Emblem to this October edition of the EMBL Entrepreneurial Minds series. Before we start, just a brief housekeeping comment for those people who are here in this room. There may be photos taken. In case you don't want this, please raise your arms now. Then we can, whatever we do, we can either skip that part of the room or we just don't take photos. So this is actually the first edition of our webinar, which is held in hybrid format. And we are glad that we have a special guest and speaker today, who is Christian Tidona. I can say it's an old friend actually of mine. We know each other since nearly 25 years. You all read his bios. I don't need to repeat what is said here. I mentioned maybe only two things. First of all, he's biologist by training. So he's, as we are all here, maybe in this audience, he started as biologist, but his heart is basically at the interface between academic science and industrial applications to create impact for the society. This is what he's working for. And Christian was, for me also personally, always an inspiration of what is possible if you want to make a difference, right? If you don't stop at the end of your bench and you really want to change things and make sure that what you do sort of helps the society. And without further ado, sorry. I hand over to Christian. His talk is he will talk about how to innovate at the interface between academia and industry. And Christian, the floor is yours. Thank you very much for joining us today.
Thank you so much, Jürgen. Thank you everyone for joining. Also everyone online. It's really a great honor to speak at EMBL. In fact, what Jürgen didn't mention, on top of the fact that we've known each other for a long time, EMBL was actually the first academic partner of BioMed, and this is how it all started. So today I'm going to tell you a little bit about, as Jürgen mentioned, about how to innovate at this interface between academia and industry, and also a little bit about what are the differences in culture between academia and industry, and what are the challenges we're all facing if we want to bridge this gap. So let's start a little bit with what are the main differences. So any ideas in the room or even online, you can put it in the chat. What do academic researchers want? What is it that we all wanted at some point and some of us switch to something else? What is it? Any idea? What do we want? Grants, that's good. Oh, money, money, money. Okay, that's good. Very, very interesting answer at EMBL. Any other ideas? What do academic researchers want? Publications. Publications, here we go. So in the end, let me formulate this differently. It's about recognition. So when all of you yes, please, Matthias.
I think what we want in academia is understanding, in the case of a biologist, phenomena. I quite honestly think that if that is your motivation,
don't stay in science. I agree. This is an observation what the majority might want. The idealistic way, Matthias Henze, just if the ones of you that didn't hear him online, Matthias Henze just said it's about curiosity-driven research and you want to find out how the world works. And if prizes is your main motivation, it's probably not a good place for you. I fully agree, but when we look at how science is organized. Most of the time, people are fighting for impact points, and they're fighting for funding, and they're fighting for the big medals. What is the, and of course, if you're a big scientist and you have great achievements, and then you sit with your grandchildren at Christmas, and then they ask you, Grandpa, Grandma, what are you most proud of in your life? And then you pull out your Nature publication, and the grandchildren look at you. What is this? But there might be other goals in life. So what do entrepreneurs want usually? Any ideas? Solve a problem. Try to formulate it in a different way. What kind of problems are these problems? It's about impact. You want to create impact in the world with a product, with a service. You want to change the world one way or the other. And these are fundamental cultural differences, what drives academic scientists and people in industry. And it's also about mindset. So you probably know this quote from Steve Jobs: the ones who are crazy enough to think that they can change the world are the ones who do. So you only need a good idea. You need to be driven to do something. But, and then if you're in the United States, you will find statements like this, like from Sam Walton, the founder of Walmart, who said, capital isn't scarce, vision is. So we have enough money, no problem, but we don't have visions. But, you know, I mean, you're probably coming from all different countries, but you might know this gentleman here, Helmut Schmidt, former chancellor in Germany. And he put it in one sentence what German culture is all about. And he said, people who have visions should go and see their doctor. So it very much depends in which environment you are. And that determines how you can change the world or do your research. Now, with a PhD in the life sciences, there are three fundamental directions in which you can move. You can stay in academia. Most of the people who finish their PhD in academia still have this career path in front of them. You can move to industry, you can start your own company, or you can do anything in between. There's lots of different jobs what you can do with a life science PhD. Now, let me try and sort of sort this out a little bit, which kind of people should go into which direction. So people who love freedom should stay in academia, because in academia, you determine what you want to focus your life on. What are the research questions you want to focus on? Nobody's going to tell you, once you're at a certain position, nobody's going to tell you how to do your research. But as I said in the beginning, when your grandchildren ask you what are you most proud in life, it's probably a very good publication where you say, I discovered something that was really very difficult to discover. But you, in many cases, you will not be involved in the development of a product which reaches people in the end. If you want to focus your life on products, on building products, then you usually go to industry. You become part of a big machinery that knows how to turn those ideas, those research results into actual products that will then reach the market. But you're an employee of a large organization, so you have no ownership. You participate, you can tell you were part of the development of this new therapy, which is saving millions of lives, but you will not get rich because you don't own it. If you want ownership, you start your own company. And then you're a shareholder. But what I can tell you from experience, and I've started quite a few companies, most of them here in Heidelberg, you don't have freedom. Once you start a company, usually you take venture capital from investors, and then you do exactly what the investors tell you. And that's the least degree of freedom you can imagine, I can tell you. So, and there's of course anything in between. And I decided that my sweet spot is at the interface between academia and industry. I wanted on one hand to be able to promote curiosity-driven research. And on the other hand, I wanted to be involved in something that generates products that will help patients in the end. And there's not many places where this really works because the cultures are so fundamentally different. But this is what biomedics is all about. So a little bit about my background. So because most of you won't probably know who I am or where I come from. As Jürgen rightly said, I did my PhD here in virology in the late 90s, so last millennium, long time ago. And And I always had two hearts in my chest, one of a scientist and one of an entrepreneur. So I started my first company during my PhD. It was called Multimetrics in Regensburg. I was the first startup in the biopark in Regensburg. And I had a very big tagline, 100 tests in 1 drop of blood. And if you followed the news a little later, there were other companies that tried to do the same thing. I didn't go to jail. But I also failed. It didn't really work. I was the first. How many of you know the Luminex technology? Luminex, yeah, of course. So it's a liquid ELISA. You put the capture molecule on polystyrol beads, 5 microns across. They have different colors, and on every color you have a different capture molecule. So you can run technically 100 ELISAs in one well. And this sounded like this could revolutionize the diagnostics industry. But at the time, I was the first licensee of Luminex. And at that time, these machines were leaky. So they had a mean time before failure of about 1 day. And then the sheath fluid was leaking, or the laser was breaking down. And also the beads had very high lot-to-lot variation. So it was impossible to build a standard diagnostic out of it. But we did cytokine assays. Some of them, similar assays, are still being run today, and also multiplex assay for borreliosis. Technically, I would call it a liquid Western blot. Then I moved to a company which already started here in Heidelberg. It was the first biotech investment of one of the founders of SAP, Dietmar Hopp. Cytonet, a cell therapy company, restructured the company, and together with the 2 managing directors, we brought the company towards combined phase 2, phase 3 clinical trials with liver cell transplants for children with urea cycle defects. It was very successful, but this was too early for cell therapy. There was no acquisition by a pharma company at some point, and then, of course, all the money was lost. Then together with Andreas Trump, I started HiStem. Some of you might know HiStem, Heidelberg Institute for Stem Cell Technology and Experimental Medicine. Together with Andreas, we were the 2 managing directors at the beginning. We organized the funding from the Dietmar Hopp Foundation, and also from the German government, and also DKFZ, German Cancer Research Center, contributed. And it's quite a successful institute, so he's still publishing 6, 7, 8 Nature papers every year. And then I moved to regional development, BioRN. You might know the local network. I actually started the BioRN company. This came out of the BioRegio competition in the 90s. We started a company that was then winning this leading edge cluster competition. And in this organization, then suddenly, with the help also of EMBL was an important partner, we had 85 million euros, and we were doing projects at the interface between academia and industry with all these partners. And then 2013, I wanted to do something different. And I was completely fed up with the, let's say, the lack of freedom you have in venture capital finance startups. What I also didn't like is that I saw many biotech companies fail miserably, not because the technology didn't work, but because at some point the investors didn't want to give more time to the science to develop, and then they pull the plug, and then everything is gone. And I said, this is not how science works. And so I started BioMedics on a completely novel concept. I'm still the owner of BioMedics. There's no investors, no public funds involved, no venture capitalists. And of course, it's not a company that grows like these biotech startups in the Silicon Valley. We grow continuously, but meanwhile, we have reached quite a solid situation. And I'm still in the position where I can say, no, I'm not going to do this project because it's scientifically not inspiring enough. It's Me Too stuff, and we don't do Me Too stuff at BioMedics. So my focus is building world-class innovation ecosystems in Heidelberg, and now we're in other places as well. Now, what is BioMedics all about? We call it 2 worlds, 1 institute, academia, industry, and 1 institute. We started on the campus of the University of Heidelberg in Neuenheimer-Feld in the Technology Park in 2013. Jürgen was one of our first joint steering committee members, and also part of the process how to shape how we could build this. It's embedded in the campus, so we are viewed as a local institute, which also helps a lot. We are at this interface between academia and industry. And because we are focused on curiosity-driven research and we publish also, we are regarded as a partner, not an industry partner, but a partner that functions very similar to academia. And we're combining the best of these two worlds, and we do this in a new way, which I published in 2015 in Nature Biotech. How does it work? So from the industry side, we get three things. The first thing is we get very, very big problems. Usually researchers in industry don't take the time, or they don't have the time to think about big problems which could be game changers in 5 or 10 years from now. They're very much focused on the current pipeline, on the problems they're solving in order to develop the current pipeline. So I usually, and that's still me in BioMed, I run workshops with at least 20 of the biggest pharmaceutical companies on a regular basis and help them to ask these big questions and then also articulate these questions in a way that academic scientists can understand what the actual problem is. Also, we get mentorship. We partner with these companies in a way that is truly a research-based collaboration. And we also receive the funding from the industry side. From the academia side, we have established a network where we have access to academic talent, to project ideas, to also academic mentors, to human samples, tissues, academic IP, and also academic infrastructure. And our first framework agreement with an academic institution was with EMBL. And we're still using this on a regular basis. And in the end, we produce what we call solutions to these big problems, and they are then transferred in an ideal situation to the pharma company, and then they turn it into something or take it as a groundwork to develop something that will help patients. And how we do this is we apply the principles of design thinking to the drug discovery and development process. So how does that work? Design thinking starts with what they call empathy, meaning, in other words, you really, really need to first understand the problem before you think about the solution. And that is what takes most of the time at BioMed, is finding these problems that are big enough for a BioMed project, that are bold enough, and where really at the moment no one has an idea how the biology works. It needs to be something which is really a big question. And that takes, in some cases, takes years with the pharma company until they come up or they agree with us on a big question. Most of the time, they're coming with these very trivial research questions. And my answer is always, if you can do it with a contract research organization or internally, do it with a contract research organization. That's not for BioMed. Once we have identified these challenges, we put them on our crowdsourcing platform and publish them worldwide at the best universities and research institutions, and invite early career scientists to apply by submitting online a very original project proposal, how to solve this particular problem. And then usually we get 200, 300, up to 500 registrations on our call, and then lots of applications. And then this is an automated system where then the pharma partner gets access. They see in their dashboard the incoming applications, and with instructions how to score them, how to rank them, and how to find the best talents. And then we agree in this system on the top 15 talents, and they enter the next stage, which is ideation. And this is where all the new proposals are developed. It's a 5-day bootcamp. So we move them to one of our sites, we invite them, we fly them in. They see each other for the first time. They're coming, super talents coming from everywhere in the world. Usually it's 13, 14, 15 different nationalities. And then we help them to associate in 5 different groups, and we help them to convert this collection of academic ideas into something we would call a truly brilliant project proposal, where you sit back and say, damn, this is elegant. Why didn't we think about this? And they present this on the last day of the bootcamp in front of a very distinguished jury, which is usually senior management of the sponsoring pharmaceutical company and us. And then we pick a winner. And this winner then gets a grant between 5 and 7 million euros for 3 to 5 years, and is moved physically to one of our institutes, where the person implements their project with very frequent mentorship, with mentors, key opinion leaders from academia, but also top researchers from the sponsoring pharmaceutical company. They meet each other once a month to discuss all the past and future experiments. It's curiosity driven. It's open science. We publish also. And we built what we call the groundwork for example, a full-fledged drug discovery program at the end of our project when we have identified and validated completely novel biology in different systems. So this is how it works. This is what such a bootcamp looks like. This was the last one here in Heidelberg with Daiichi Sankyo. The question here was, how can we target solid tumors with bispecific or multispecific biologics in a way that the proximity induction that is the result of this bio multispecific molecule will trigger a completely novel pathway which was not characterized before. So bringing molecules together, for example, on the surface or inside of tumor cells or tumor-associated cells in a way that triggers, in the end, the killing of the tumor. So novel biology, novel chip technologies. That's a typical biomedics project. Good, a little bit of history. Meanwhile, after 13 years, I think I can say we're a leading innovation hub for pharma. We've been working with 13 different pharmaceutical companies and also recently with the Gates Foundation. And our mothership is still here in Heidelberg. We're in the technology park and we're moving next year, finally, because we are in different buildings, we're moving into the same building and we're increasing our space from currently 1,800 square meters to up to 4,000 square meters of lab and office space. We've started 26 projects in the past 12 years in Heidelberg. So do the math, each of these projects is roughly 5 million. These companies have invested far more than 100 million euros into our projects here in Heidelberg alone. Since last year, we have the very same concept also very close to the campus of Yale University in New Haven, Connecticut. That's actually where we'll be flying to on Monday. And we have a local team. We have the first research group on site, and the next project is about to join early next year. And we have the same ties with all the academic institutions locally like we have here in Heidelberg, especially, of course, with Yale University. Then we've started a completely new model. It's called Exceed Labs. The question from pharma was, can't you put this inside of our campus? And that's what we did in Boehringer Ingelheim in Ridgefield, Connecticut. It's a large pharmaceutical campus with really, really nice core facilities, latest technology on site to do great research. And now we're operating an incubator inside of Boehringer Ingelheim. All the scientists are on our payroll, but they can use everything. They can sit in any scientific meeting of Boehringer Ingelheim. They can sit in our meetings. They can use the core facilities, the recreation facility, the restaurants. They're like Boehringer Ingelheim employees. And the second project is about to join. We just successfully finished the second bootcamp there. And it's now being spread. So next pharma company wanted that as well, which is Servier in Paris. So we are starting a new Exceed Labs incubator inside of Servier in Paris-Saclay early next year. Bootcamp is still this year. And hopefully we're going to put that in as many pharma companies as possible because it's a plug and play thing, right? So our processes are very simple to implement for a local team. Then I'm going to tell you a little more in the last 5 minutes about one of our newest achievements, which is we won a major tender from the Gates Foundation, $10 million. And it's about building based on our model, a women's health R&D accelerator here in Heidelberg inside BioMedX, which focuses entirely on women's health problems, especially with applications in low and middle income countries. And this is, as Jürgen said, this is very close to my heart because in the end, it's about generating impact. And in this case, having the very strong partner like Gates Foundation on board, and then being able to focus entirely on things that are really needed in those low and middle income countries is very rewarding. And similar project also about impact. This is in Barbados. This is sponsored by the Prime Minister's office in Barbados and also the European Union. It's so much in focus that in February, when we signed the agreement, there was the President of the European Commission, Ursula von der Leyen, in Barbados visiting us together with the Prime Minister. And then we signed the documents. And what we're doing here is it's very similar to women's health, right? As you might know, most therapies still today are developed with clinical trial subjects who are white males, middle-aged. But the world's population is very diverse. So half of the population is women, is usually not taken care about in clinical trials. And then old people, children, people of all ethnicities. So something that works in middle-aged white men doesn't necessarily work in all the other groups as well. And the question is, how can we use AI and deep molecular phenotyping of patients to build models where we can adapt those therapies to the other population groups? And in Barbados in particular, there's problems with cardiometabolic diseases. If people have been to the Caribbean, most of the population locally is of African descent. And they all have obesity problems, or many of them, and also cardiometabolic diseases. And the interesting thing is that the patient-to-patient variation of these cardiometabolic treatments is very high there. And that's probably based on genetic background. So we're working on that. Of course, you know Heidelberg, Neuenhammerfeld campus, and then also satellite here is the EMBL campus, a very dense research area. And we're also collaborating, meanwhile, with more than 60 academic institutions worldwide. Our R&D focus and expertise is in 3 main areas. One of them is building really relevant in vitro models of human disease. Almost all of our teams are receiving primary human materials, some of them whole human organs from multi-organ donors. And then we build our own chip layouts and very complex disease models where we include into the different cell types and tissue types. We include vascularization, we include the immune compartment to be able to see something like COPD developing in a dish or fibrosis developing in a dish, which is very complicated to do. And then almost all of the teams have bioinformaticians or AI machine learning experts, since we're producing lots, like also at EMBL, lots of very deep data sets. And also some of the teams are working on really physical sensors, like optical sensors or electrical sensors. And you have to imagine that all these teams, every Friday, we connect them to each other at all the different sites. The group leaders decide who of their team members, PhD students, postdocs, presents their work or failed experiments in front of everyone, and everyone contributes how to make it better. And then you have an immunology PhD student in front showing a failed experiment, and then there's the sensor person who says, oh, we have this little sensor, you put it in your well, and then you can measure online. Or there's from the bioinformatics team, there's someone says, oh, there's this new algorithm from MIT, let's tomorrow sit together and reanalyze your data. And they were all selected through this very rigorous call and bootcamp process. So really top of the top. Good, this is our platform. We publish here all our calls for application, all our job offers at all our sites. Currently, there's over 14,000 actively job-seeking researchers in the system from 140 countries. And what is most important at BioMed is our core values. And this goes back to the starting moment of BioMed. Everything is flexible at BioMed, but not the core values. We're not discussing. And if you come and visit, you'll see them in almost every room. The first one is cross-leverage. We inspire and motivate each other to unleash our full potential, meaning BioMed is not just a place for great scientists, but great scientists who have embraced the concept of growing by helping others. This is why we are doing these boot camps. We don't let people sleep for 3 nights in a row. And if great scientists turn to jerks, even if you have 20 Nature publications, they have no chance of joining BioMed. Cross-pollinate, we embrace diversity as it's the source of innovation. No, this is one of my key mantras and my learnings also in all the different companies, is that the more people look upon the same problem that are coming from different perspectives, the higher the probability that something really elegant and innovative will come out. So it's very important that we have people with very different views. Of course, they all need to be great scientists, but they come from all different cultures, from all genders, from all religions. And while everywhere in the world we're having wars, the people from countries that are usually at war together, they are side by side in our lab, and they're collaborating very productively and helping each other. And aim high. Our goal is to enable a new generation of targeted therapies. So BioMed is not a place for a quiet 9-to-5 job. We're putting a lot of effort in providing all the resources that's in that respect is very similar to EMBL, so that you have everything you need to be very creative as a scientist, but we expect you to make most out of it. And BioMed is also, like very similar to EMBL, a temporary place. Our maximum is 5 years, at EMBL is 8. And then you need to move on, because we believe that for scientists it's important to get to the next step. About 70% of our fellows go to industry, they transition, they use BioMed as a transition to industry, and about 30% go back to academia, because we also publish, and they have a continuous publication record, and then they become professors. Now, finally, a topic which is starting right now and which might be interesting for one or the other person in the room or online who are thinking about the next step in their career. This is this Women's Health R&D Accelerator. And the way how it works is that we get for every project a seed financing of $2 million from the Gates Foundation. And this means we can really concentrate on very basic problems. Then we have, in this case, mentors who are not just mentors from a pharma company that is sponsoring, but mentors who are really global. They come from top companies, institutions worldwide that help the teams to develop something really transformative. And then we generate multiple projects with this money. And the goal is after 2 years, after a major milestone, that an additional funding partner steps in and continues financing the project. It can be investors, then we turn it into a startup. It can be a pharma company, then we turn it into a normal BioMed project, or it can be a public funding agency or a bank even, and then we find a solution as well. And this is just starting, so we are trying something new, like always at BioMed, and let's see where this is going. Our first call in this series, we call it XFAM Labs. So this is a Women's Health R&D Accelerator, is new strategies for female-controlled non-hormonal contraception. And you can imagine that if there was something like a pill that does not interfere with the female menstrual cycle, that could be very helpful, and especially in low and middle income countries, and in countries, for example, where you have, in Muslim countries, where the female cycle is very well observed by everyone, and you have a problem if you mess with your own cycle as a woman. In this case, women would be in control. They could control their contraception without anybody noticing. So that would sort of liberate women in many of these countries. Major objectives here is identification and validation of innovative non-hormonal mechanisms of action, so new biology, demonstration of this contraceptive efficacy in vitro and in vivo. The question is, how do you model female reproductive organs in vitro in a way that you can make a meaningful assumption about the efficacy in the human? The launch of the call is on Monday, and this is the link. This link is not yet active, so you're the first ones to see it. It will go live on Monday. And with that, I would thank you for your attention. It's really a pleasure, as I said, to speak here at EMBL, and I'm very happy to get your questions. Thank you.
Thank you very much, Christian, for this excellent talk. I hope you agree that, again, it's inspiring of what Christian does and what he triggers. And maybe just an anecdote. I had the privilege in the first years to participate in the steering committee or the juries at the end of these boot camps was described. And it was, as he said, it was amazing to see the level of excitement of these students, of these people, these scientists. I had the privilege to come the last day, more or less, right? And I sensed a mixture of super excitement, and they were nearly dead because they come, of course, to the boot camp. Their project proposal is more or less dissected and destroyed in a positive sense. Yeah. But over the weekend, they work like hell. They don't sleep. And Christian manages really to keep the attention, the spirit, the engagement at the highest level. And the people are just great when they present, when they pitch in front of the jury. I was always very impressed to see how successful also these projects have become.
And they stay connected. That's the interesting thing. Yeah. I mean, it's being together in such a boot camp, going through all of this gets people together. And what we're seeing is that these alumni are still working with each other even 10 years afterwards. So even the ones that were not chosen were benefiting from this program.
Maybe I kick off the discussion with a question. The exceed concept. Why did Boehringer Ingelheim ask for it? What is the advantage from a certain perspective, and how do you see it? Isn't there a risk that what you achieve in the BioMed Institute is sort of a bit softened, and by implanting these teams now into the company, suddenly they can be soaked into the regular pharma sort of machine, regulatory procedures, processes, and so on and so forth?
Very good argument, Jürgen. That's in fact what needs the most energy, to make sure how we manage these teams, that they're not sort of being gobbled up by the structure inside the pharma company. Why is this needed? So if you look at pharmaceutical companies, all of them know that they need diversity on their R&D campus, and diversity means people from outside, especially academic people. And they have all tried to put people from the outside inside their campus. And there is 3 things that make it very complicated. One of them is IP ownership. So if they're still employed at a major institution, who owns the IP in the end? Second is confidentiality. They will get to see all kinds of confidential things if they talk to the local people. And third is labor law. So if you're local on such a campus, in many countries, you need to get the same salary, for example, and you need to obey the same rules. You cannot have overtime and so on. And all the pharma companies, I can say all the pharma companies have failed in trying to put these academia-like models inside their campus. The only way how they solved it is putting red tape around the groups. They're not allowed to talk to anyone, not allowed to mix with the other people. And then this whole thing doesn't work anymore. It's like, and we had the same situation when we discussed with Boehringer Ingelheim. So their motivation was, can you organize it for us in order to increase our innovation productivity at our site? And we said, yes, we try. And then we sent over an agreement proposal, and it came back 3 times as long and red from the legal department. And they came with this red tape, right? They cannot leave this, and they cannot do this, they cannot this. And I said, well, it's like, it's like being pregnant. You're pregnant or you're not pregnant. You cannot be a little bit pregnant. And in this case, this doesn't work. So they need to be free. They need to communicate. Otherwise, it doesn't make sense. And then senior management, the CSO, Michel Perret, said, make it happen. And then we got the agreement that actually enabled us to do this. And now we have a precedent, and now other pharma companies follow with the very same agreement structure.
Right.